Okay, so we start with the microvilli. Okay. So microvilli, they are microscopic cellular membrane protrusion. Means that it's something like a finger-like projection come out from the cells. So if I draw it, it will be something like this. Can you see that? It's like a finger-like structure that actually come out from the cells. Okay. So microvilli, if you look at it, what is their main function? Their main function actually highlights just to increase the surface area, particularly for transportation. When we deal with the absorptions of the nutrient. When I say like this, then you know that microvilli, where we can find them? Definitely in our small and large intestine. Small intestine absorb the nutrient, large intestine absorb the water molecule. So we increase the surface area. Another one is our kidney, when we want to have the selective reabsorption, right? For the glucose. If no glucose will appear in the urine, everyone also will have the diabetes. Correct or not? Okay, so we want to reabsorb back all the glucose in the PCT cells. So there are the places that we were going to get the microvilli. So what is actually inside this microvilli? Okay, so if I zoom into these microvilli here, we can find out that they consist of cytoplasm and microfilament. Are you clear? The cytoskeletons, microfilament, not microtubule. That's why we learn microfilaments before that. Are you clear? Okay, we learn microtubules, now we learn microfilament. So it means that at these regions, you will see a lot of these microfilaments because you have to make sure that the structure won't collapse, right? You try to imagine the house without the pillar, cannot. So the organism without sorry, for example, mammal, animal without the backbone or without the exoskeleton, you must have a skeleton there, right? So in this case, this part, you can get cytoplasm as well as microfilaments. But be careful, even though we call it cytoplasm, there is no or very little, no cellular organelles that present in the microvilli. Yeah, okay, highlight no cellular or very, very little and no. Okay, the microvilli. And each microvilli has a denser bundle cross-linked actin filaments. Actin filaments also belongs to microfilament. If you forget about this term, it's okay. Okay, you just term it as microfilaments. It's correct. Okay, so thousands of microvilli then form this something like a brush border. It's something like a brush. So it's found on the apical surface of the some epithelial cells, such as the small intestine. Okay, now what's the apical surface? Apical surface is the one that faces the lumen. So let's say this is intestine. So we get the cells here, you can see that the cells will have the microvilli like this. Okay, so this one is the lumen. Are you clear? Okay, so this is a cell. So the part, the surface that faces the lumen is called apical surface. This surface is known as apical surface. So that's why we need to increase. We need to increase the surface area. And this microvilli membrane sometimes also packed with the enzyme that digests the carbohydrates. Okay, so this enzyme is termed as glycosidases. Glycosidases actually is the enzyme that break down carbohydrates. So in small intestine, we need to break down the maltose. We need to break down the lactose. We need to break down the sucrose. Cannot. So we have the sucrase. We do have the maltase. Those enzymes, okay, we don't secrete them. We place them onto the membrane. So therefore, you can see that we have enzyme and the membrane. So then we increase the surface area, we can accommodate more enzymes to carry out the functions. Okay. So if you check the diagrams, okay, you can see that this is the microfilaments. Okay, F, okay, here stand for actin filaments or microfilament, and you won't see very basal body, guys. Cilia, you will get basal body. Here you can see that no basal body. Okay, so this is the longitudinal sectioning, but for transverse sectioning, even easier for us to identify. Why? Do you realize that if we compare to cilia, cilia have 9+2 arrangement, but microvilli, they don't have this arrangement. So it's scattered. Can you see that? The microfilaments, they are scattered. So this is how we identify between the cilia. Be careful when I place two diagrams to you guys, you need to identify them. Which one is cilia, which one microvilli? Because question may say that, okay, refer to the figure, okay, state the functions of this structure. Then you write cilia. Cilia, different function. Okay, microvilli, different function. So means that we need to know how to identify the structure. So transverse longitudinal sectioning a little bit hard, okay, for us to actually look at it. So what we look at it here is the microfilament continue. Can you see that? The microfilament continue. So this one is the microvilli. If cilia, then it's no. You will have the basal body. Okay, cilia, you will see basal body. Okay, which act as the M tox. Okay, still remember. And then for the transverse sectioning, very simple, even easier for us to identify due to the microfilament that's scattered. Okay? You won't see 9+2 arrangements. Are you clear? And cilia, they are non-motile. So cilia, the microvilli, they are non-motile. So means they won't move. Okay, they don't move. So you won't see them bend. Okay, can you see that? You won't see them bend compared to the cilia. You check back the cilia, the notes, they will bend, right? This one, you won't see them bend because they are non-motile. Okay. So comparison that we need to know here, microvilli versus the cilia. So microvilli location, microvilli mainly occur at the epithelial cells of the small intestine and the kidney tubule. Okay, so they involve more to the reabsorption. So microvilli increase the surface area for absorption or transportation. Okay, so how about in terms of structure? So microvilli are made up of microfilaments. I highlight microfilaments. So therefore, they lack of 9+2 arrangements of microtubule. They don't have 9+2 arrangement of microtubule. But cilia, yes, cilia are made up of microtubules. Highlight, they are microtubules. So, but in this case, the microtubule also arranged in a very special order called 9+2 arrangements compared to the microvilli. In terms of motility, definitely microvilli, they are non-motile, but cilia, they are motile. Motile doesn't mean that they can move, but the beating action can help us to move the particles. So where we can get the cilia? Cilia, basically, you can see that at the respiratory tract. And we have learned this before. For example, in our trachea, in our bronchus. Are you clear? And bronchiole, you can see the cilia. Or uterine tubes, okay? Or we call it fallopian tube, where this cilia, the beating action of cilia can move the secondary oocyte, okay, towards the uterus, or actually sweeping this embryo to the uterus. So this is microvilli, okay, compared to the cilia, right? The differences. So people with celiac disease, okay, it's a special type of celiac disease, have the immune response to gluten. Okay, nowadays people actually go for gluten-free food, or okay, why? Because glutens, it may act as an allergic reaction. So immune response actually targets these glutens and damage the microvilli. So when it damage the microvilli, means that the intestinal cells cannot absorb nutrient normally, leading to what we call the malnutrition, cramping, and even the diarrhea. Okay, so we're going to learn this one. Okay, talking why if we cannot absorb the nutrient, we will get diarrhea. Okay, in next chapter when we look at the membrane and transport, okay, across the membrane. So this is not in the syllabus. Okay, just for information only. Celiac disease. Celiac disease is a special case where our immune system target the gluten. When it target the gluten, they damage the microvilli of the small intestine. So therefore, you lack of the surface area. Can you see that your surface area becomes smaller already? So therefore, your ineffectiveness in terms of the absorption of the nutrient, leading to the malnutrition, cramping, as well as diarrhea. Okay, so with this, I stop the recording now.